Welcome to this exciting lecture on the applications of biostatistics in biological sciences. Today, we are going to explore how mathematics acts as the universal language of modern biology. We will see exactly how raw, unpredictable biological data is transformed into meaningful, life-saving discoveries. Whether you are studying zoology, medicine, or ecology, this foundational knowledge is absolutely essential. Let us dive right in. Now that we have introduced our topic, let us start with a simple analogy. Imagine you are a detective trying to solve a mystery in a massive forest. You want to know if a specific berry is making birds sick, but you cannot possibly check every single bird. Instead, you observe a small group, record their diets, and look for patterns. This is exactly what biostatistics does for scientists. It provides a set of mathematical tools that allow us to take a small amount of messy, unpredictable biological data and uncover the hidden truths within it. Without these tools, we would just be guessing. Building on that detective analogy, let us look at why biology specifically needs these mathematical tools. Biology is inherently full of natural variation. No two cells, plants, or humans are exactly identical. Biostatistics is the specific branch of statistics that quantifies this natural variation. By doing so, we can clearly distinguish between random chance and actual scientific effects. By applying mathematical principles to biological data, researchers can draw objective, evidence-based conclusions rather than relying on subjective guesses. It bridges the gap between raw numerical data and meaningful biological discoveries, ensuring our scientific claims are reliable and trustworthy. Understanding how we manage variation leads us to see the immense real-world impact of this field. The importance of biostatistics cannot be overstated. It is the backbone of almost all modern medical and biological research. Without it, we would have no way to prove that a new cancer drug actually saves lives, or that a specific vaccine is safe for the public. It is heavily used in epidemiology to track infectious diseases, in genetics to determine inheritance probabilities, and in agriculture to maximize crop yields. Without biostatistics, research would rely entirely on anecdotal evidence, leading to dangerous decisions. Now that we understand the broad applications, let us focus on a specific use case in epidemiology. One of the most common applications of biostatistics is measuring how common a disease is within a specific population. To do this, we calculate something called the prevalence rate. This metric tells us the proportion of individuals who have a specific condition at a given point in time. This simple but powerful calculation allows public health officials to understand the true burden of a disease. With this information, they can allocate medical resources accordingly and make informed public health policies. This concept of measuring disease burden brings us to our first mathematical formula. The prevalence rate is calculated by dividing the number of cases by the total population and then multiplying by a standardizing factor. Let us break down the symbols. P represents the prevalence rate we are trying to find. C stands for the total number of existing cases of the disease. N represents the total number of people in the population who are at risk. Finally, 10 to the power of k is a multiplier, like 100 or 1,000, used to make the final decimal easier to read. If the number of cases increases while the population stays the same, the prevalence will increase. Before we move on, let us see this formula in action with a concrete example. Suppose a town has a total population of 5,000 residents, and doctors discover that 250 of them have a viral infection. We want to find the prevalence as a percentage. First, we identify our given values and substitute them into the formula. We replace C with 250 and N with 5,000. We use 100 as our multiplier to get a percentage. Dividing 250 by 5,000 gives us 0.05. Multiplying by 100 gives us 5. This means exactly 5% of the town currently has the infection. Having seen how biostatistics tracks diseases, let us shift our focus to another major application: agriculture. Biostatistics is heavily applied in agriculture to evaluate the effectiveness of new farming techniques. Imagine a botanist is testing a new organic fertilizer on a specific species of tomato plant. They need to know if the fertilizer actually worked. To do this, they want to find the average, or mean, weight of the tomatoes produced. By finding a reliable average, the botanist establishes a baseline. This baseline can then be scientifically compared against tomatoes grown without the fertilizer to prove if crop yields truly improved. This need to establish a reliable baseline brings us to our next mathematical tool. To find that average weight, we rely on the fundamental biostatistical formula for the sample mean. Conceptually, this involves adding all individual values together and distributing that total evenly across the sample. The symbol X bar represents the sample mean we want to calculate. The sigma symbol, followed by X sub I, tells us to take the sum of all the individual data points. The variable n represents the total sample size, or the number of items we measured. If we increase our sample size n, our calculated mean generally becomes a much more reliable representation of the entire population. Now that we have our sample mean formula, let us apply it to our botanist's tomatoes. The botanist harvests 4 tomatoes, weighing 120, 130, 140, and 150 grams. We plug these specific biological data points into our formula. In the numerator, we add the 4 weights together. In the denominator, we place the number 4, representing our sample size. Adding the numerator gives us a total combined weight of 540 grams. Dividing this by 4 distributes the weight evenly, resulting in 135 grams. This tells us the average weight of a fertilized tomato. Before we visualize the entire process, let us discuss the standard workflow that every biostatistician follows. Every application of biostatistics begins with a clear biological problem, such as an unexplained disease outbreak or a need for better crop yields. Once the problem is defined, researchers move to rigorous data collection, ensuring they sample the population correctly. After the data is gathered, the core biostatistical analysis takes place, utilizing the formulas we just learned. Finally, this mathematical analysis translates directly into a scientific conclusion. This structured workflow is what prevents researchers from jumping to false conclusions based on limited observations. Now that we understand the steps conceptually, let us look at this workflow visually. What you are looking at is a flowchart that maps out the entire journey from a biological problem to a scientific conclusion. On the far left, we start with a biological problem, such as a disease outbreak. The first arrow leads us to data collection, where we sample the population. The next arrow takes us to biostatistical analysis, where we calculate metrics like prevalence or mean, just as we did in our examples. Finally, the last arrow points to the scientific conclusion, where the analyzed data is used for resource allocation or treatment approval. This diagram perfectly summarizes how raw data becomes actionable knowledge. We have covered a lot of ground today, So let us summarize the most important ideas from this topic. First, biostatistics is the essential tool that transforms observational biology into a rigorous, quantifiable science by cutting through natural variation. Second, mathematical tools like prevalence rates and sample means allow us to find true, reliable patterns in messy biological data. Finally, whether you are tracking a global pandemic or improving agricultural yields, biostatistics provides the absolute framework for making evidence-based decisions. You now understand the foundational applications of this field, which is the first major step toward mastering biological research.